Welcome. Today we are going to learn about Vis-SPLIT, an interactive clustering framework designed to build classification models for different cancers by observing genetic input data. RNA sequencing is a process of collecting and measuring the RNA molecules in a biological sample. Measuring the abundance of these molecules allows us to measure the expression of different genes, which holds diagnostic value in the classification of cancer and prognostic value in the understanding and prediction of the course a disease will take. Existing classifiers, such as the PAM50 system, were formed from labor-intensive manual hierarchical clustering, which has made it increasingly difficult to maintain and update them, even when provided with more data and modern technological advancements. The approach to analyze and visualize RNA sequence data has been to use dimensionality reduction techniques. More recent nonlinear techniques, including machine learning methods, have found some success differentiating diseases based on genetic makeup, but these black box techniques lack transparency and interpretability, which is crucial in the classification of cancer. Linear methods draw direct links between input data and resulting classification, but they might not capture all the important patterns in a single pass. These methods are also often manually configured to a specific dataset, keeping them from becoming generalized classification models. To address these issues, we propose a novel interactive clustering and modeling tool, as well as a case study evaluated by two domain experts. Namely, we present the tool Vis-SPLIT. Powered by iterative execution of principal component analysis, this tool allows a researcher to build a classification model by focusing on one identifiable genetic distinction at a time. This tool is made up of a few linked views. The hierarchical overview will show the current state of the model and update as the analyst progresses. The heat map overview will reveal the genetic signatures of clusters as they are formed. The survival analysis view allows comparison of prognosis between different clusters. The PCA view provides analysts with projections, which they can analyze, manipulate, and ultimately use to define the model's partitioning decisions. In addition to the projection, this view contains two aligned heat maps and a loadings plot. When given a projection, the analyst will first want to know how different genetic features are influencing it. The loadings plot can be used to explain why certain visual separations are apparent in the projection and can also indicate possible correlations between similarly contributing genes. Additionally, aligned heat maps are provided next to the projection, revealing the genetic expression across the X and Y axes. This not only enforces the identification of correlating gene groups, but can also explain genetic trends when visual partitions are less clear. Before making any partitioning decisions, the user can first manipulate the projection as they see fit. One example of this is by selecting certain features by brushing in the loadings plot and then re-executing PCA on the reduced set of features. This may be done when the researcher would like to temporarily remove some features that do not seem to contribute to a noticed visual partition. Features may also be selected within the axis-aligned heat maps based on some observed pattern or correlation. Users may also encode color to the projection, revealing the expression of the selected genes, which can further assist their decisions. Once the analyst is ready to define a partition, they can choose a dividing line across the projection. This partition updates the linked overviews. The updated model is represented in the hierarchical overview, with the newly formed distinction being explained by specific genes that were automatically determined to be significant. Similarly, the heat map overview shows the new clusters and their genetic signatures, and is reorganized based on the decision. Clusters of individuals, shown as columns, are separated based on the partition, while the identified groups of genes, shown as rows, are separated and pulled to the top of the heat map. As the analyst continues to partition the data into smaller clusters, the differences in their genetic signatures will become more clear. In addition to the heat map overview and hierarchical overview, the survival analysis view is also updated with each partition. This view performs Kaplan-Meier analysis on each of the clusters to produce survival curves, which reveal the prognosis of different diseases as they relate to the clusters. A baseline curve is also given to represent the entire dataset without any clustering. The resulting model is shown as a decision tree-like structure. Looking at the tree from the top down, there are nodes that represent partitions between the different clusters, each formed from one of the researcher's decisions. The significant genes identified before are embedded in this structure, allowing the model to be easily understood. As a case study, two biomedical researchers from the Memorial Sloan Kettering Cancer Center used Vis-SPLIT, looking at the Pan-Cancer Atlas dataset for breast invasive carcinoma. They were interested in how easy it was to notice patterns and create a decision model. They first identified the more obvious HER2 and basal groups, and then noted that while there wasn't a clear division for subtypes within the luminal group, it was a less significant distinction to make, and they could still partition based on higher expression of certain genes. The result of Vis-SPLIT partitioning was a set of more clearly defined clusters, whose genetic signatures were more cohesive than with the previous PAM50 classifier. Additionally, the previous classification left many individuals labeled as none or other, even though their genetic signatures may align strongly with the defined clusters. Through our case study and evaluation from biomedical researchers, it is shown that Vis-SPLIT is easy to use and quick to identify important distinctions in genetic signatures. The researchers were able to easily extract clusters resembling the HER2, basal, and luminal groups defined by the existing PAM50 system, and could easily see genetic trends relating to the subtypes of the luminal group. As compared to the PAM50's classification of the same dataset, genetic signatures were more clearly represented in Vis-SPLIT clusters, and the domain experts noted that outputted Vis-SPLIT models could provide future researchers with quick and easily understood classifications that could be reduced further into simpler decision models if desired. Thank you for joining us to learn about Vis-SPLIT, a novel framework for interactively clustering RNA sequence datasets. Medical researchers expressed that our tool was intuitive to classify their data and produced an easily interpretable classification. We believe that it can also help other cancer researchers in the exploration of data, allowing them to build new models and compare them with existing baselines.